Well, I'd like to thank the organisers for the opportunity to speak. I'd like to describe to you some work I've been doing on super-resolution fluorescence microscopy, which I think is a very exciting topic that has a lot of potential. As I'm sure those of you with more biological training than myself will notice, podosomes are not, in fact, a structure in neurons. In fact, there's almost nothing about neurons in my talk. But I think the method does have potential for application to neurons, and we'll just have a little flavouring at the end. So, super-resolution fluorescence microscopy has really exploded as an area over the last 10 years. It's driven down the resolution you can achieve in a fluorescence microscopy experiment by up to a factor of 10. And it's shown tremendous promise in terms of observing previously unknown cell biology. However, there still may be a critical mismatch, I think, between our ability to uncover new cell biology and our ability to develop new technology. There's this fantastic range of new technology out there, but applying it to uncover really interesting new biology is an area we're stumbling on slightly. So I hope that the techniques I'll present to you today will maybe present some new possibilities for your research and maybe some solutions. So when I started in this area around 4 years ago now, I thought this super resolution sounded like a really exciting field. But of course I wanted to collaborate with cell biologists to answer some interesting questions. I went round to them and asked what they thought were the most important elements of a technique to answer important biological questions. And I came up with a wish list, which very much drives my own research. They told me that the ideal technique would be high resolution in X, Y, and Z. It would be rapid, so that you could look at dynamics. And of course, also, it would be low enough phototoxicity that you could use it in live cells. They would also like it to be able to be used with existing fluorescent proteins. Otherwise, you have to make you have to transfect again, and that's a process that can go wrong many times. They don't tend to want to buy an expensive microscope. As with all of this in these hard economic times, you don't tend to have an extra $600,000 lying around to purchase a brand new shiny system. And finally, if you have to do any post-processing, they prefer it to be real time. And I'm pretty sure at the end they said they'd like a pony as well. So there are really three major methods which are currently used to achieve super resolution. And I'm not going to go into detail how they do it, but each of them uses nonlinearity in terms of excitation or emission to boost the resolution achievable below the diffraction limit. And the diffraction limit in terms of the wavelengths that we're going to be using for fluorescence microscopy is around 250 to 300 nanometers. So the three methods that we currently use, firstly, you can use a confocal type system where you scan a focused beam and make the effective size of the incident beam smaller. And this is called stimulated emission depletion. Secondly, you can project patterns onto a sample in a wide field microscope, which downshifts high frequency information. And then you can use a more sophisticated deconvolution to re-access that high frequency information in a reconstruction. That method is called structured illumination. And finally, instead of doing this, you can use the time, and we've already heard in one of the talks yesterday about achieving spatial resolution from time. This is a slightly different use of the same concept. You separate the fluorophore emission over a longer time, over a number of images, and then you reconstruct one image from those data. And in fact, this has an almost infinite number of acronyms to name it, since every single person in the field has their own name. Storm, Palm, Resolved, and so on. But I'm just going to call it localization microscopy. Right, so how does separating fluorophore emission in time help us? So the resolution in fluorescence microscopy is limited by the size of the point spread function. Now, the point spread function, of course, is the shape which is made by imaging a point source. And if we look at the cut in intensity along the x direction, you can see it has a finite width. And this width is your resolution. It's a limitation in your measurement. However, if we know that this image is coming from a point source, we have one extra piece of information, and that is that we know what shape this should be. We know what the point spread function of our microscope should be. So, we can perform a fit to this peak, and this here is shown in red, and then the accuracy with which you know the position of that single fluorophore is limited by the error between your fit and the data. And I think you can see that the error between the red line and the blue line is much, much smaller than the width of the peak. In fact, it's different by about a factor of 10. So what does this translate to in terms of data that you'll actually see in the real experiment? Well, this is a standard example of localization microscopy data. You can see in each frame, the fluorophores are fairly well spaced. There aren't many overlapping. In this case, this is achieved by using photoswitchable fluorophores. These are a particular class of fluorophores in which one wavelength of light can be used to switch the fluorophores into a non-emitting state, and the second can be used to illuminate and make the small number that you've put into the emitting state emit light. So, to perform a reconstruction, we would threshold each one of these several thousand frames, localize the positions of the fluorophores in each of the frames, and then, like in an impressionist painting, build up a picture from a series of spots. And in this case, you'd get a picture something like this. So, this method works very well. It's relatively accessible because in addition to using photoswitchable fluorophores, you can also use standard organic dyes that I'm sure many of you will have used in your experiments, things like the Alexa dyes and the Atto dyes. And with the addition of a strong reducing agent, those can be made to blink in the way you need to do a reconstruction. However, there are several limiting factors to this type of experiment. Firstly, of course, I'm sure you'll have spotted that that data took quite a long time to acquire, so you're looking at several minutes. And of course, if you did want to use organic dyes, putting large amounts of reducing agent is unlikely to do much for a live cell. So, acquisition time. How can we improve that? The basic thing you need to be able to do to acquire faster is to be able to look at more fluorophores in each image. If you look at more fluorophores in each image, then eventually you're going to get to a point where some of them overlap. And in that case, you need an analysis method that can cope with overlapping fluorophores. Secondly, blinking fluorophores reappear multiple times, sometimes separated by periods where they're not in the image for several frames. If you can link all the reappearances of a fluorophore, your resolution will be boosted because your resolution is determined by the total number of photons from each fluorophore. And finally, you want to be able to use as wide a range of fluorophores as possible. The better your data analysis, the better the possibility of coping with poor quality data and low photon yield, and widening it to a range of fluorophores is actually useful to a biologist. So our method to do this was, instead of thresholding each frame and localizing individual fluorophores, to consider the data as a whole, as one data set, and to make a model which describes the data as arising from a number of fluorophores. And I'm just going to quickly run through a little bit of the maths. I think there's only one equation, so I hope it will be reasonably accessible. So let's consider firstly, the simplest possible case, where we're only looking at comparing two possible models, a model where we don't have a fluorophore and a model where we do have a fluorophore. And I want to do this because it's the easiest way to think through the Bayesian statistics that we're going to be using. So the key thing in Bayesian statistics is you have to compare two different hypotheses. And in this case, we'll be comparing the null hypothesis. I'm sorry this has come out a little dark for the level of lighting, but this is just a series of images of Gaussian noise, which is, or you could also model it with Poissonian noise, which would be your null hypothesis that there is no fluorophore. And secondly, the hypothesis that there is a fluorophore. And here I've varied the different parameters that the fluorophore could have. It could have different brightnesses, sizes, positions, and so on. Now, what we want to do our calculation and compare the model evidence for each of these hypotheses is we need to integrate over all the parameters. Now, there are two types of parameters or variables here, the discrete and the continuous. The discrete variables are the blinking sequences that we observe. So to give the very simplest example of the calculation that we're going to be doing here, this is an example for one fluorophore appearing in two frames. And when we integrate, what we just do is add up the probabilities. So we add up the probabilities that the fluorophore is on-off, off-on, and off and off. And that method is called the forward algorithm. Now, the continuous variables, of course, you can't just add up because they're continuous. So instead, we need to integrate. And to do that, we use Laplace's approximation, which approximates the distribution of all the variables as Gaussian. And in order to use that, we just need three pieces of information: where the maximum likelihood is, how wide the peak is, and how high it is. Now, in fact, of course, we don't have one fluorophore. We have many fluorophores. And we have to build more than one fluorophore into the model. And in fact, this was very simple. And we got to this point several years ago, and we thought everything was going wonderfully. And then we started introducing more fluorophores into the model, and we realized that, of course, this sort of model is exponential in the number of fluorophores, and therefore it would take until the heat death of the universe to do the calculation for 20 fluorophores. And so unfortunately that wasn't very useful. So we experimented with other types of calculation, and eventually we drew samples from the state sequence using Monte Carlo Markov chain. However, this did cause problems with optimization, and so eventually we came to a method where we optimized one spot at a time. For that spot, for that fluorophore, we integrated using the forward algorithm, and for the surrounding fluorophores, we integrated using Monte Carlo Markov chain. And because then the error in the Monte Carlo Markov chain sampling is on a smaller perturbation, it's small enough that we can carry out our optimization accurately. And the final stage in our analysis was realizing that each of these Monte Carlo Markov chain samples is a valid statistical sample, and therefore you can use each step of the optimization as a valid sampling and simply build up the picture by resampling over and over again. There is a large enough number of state sequences that you're not going to resample the same ones over and over again. And this is an example of a circular structure where on the left you can see each Monte Carlo Markov chain sampling, and you can see each sampling is fairly inaccurate. It has a lot of noise, but when we build up the samplings over a large number of tries, we get a very, very accurate picture. So the test structure that we chose to look at is called Podosomes. These are a type of cytoskeletal structure that formed the cell membrane, particularly in macrophages, they consist of an actin core around 500 nanometers in diameter, surrounded by a ring of actin-associated proteins such as tailin, vinculin, and paxillin. And here, on the right, we can see a cut through a podosome. In the center, you see the actin core, and here, the ring of actin-associated proteins. And I think this cut through gives some insight into why podosomes are such a very attractive structure for doing super resolution microscopy, because this ring is very, very thin, and Z resolution in super resolution techniques is still evolving. It's not really getting into commercial systems yet. They're just emerging, the first commercial systems. And it's much more challenging. If you're developing a technique, you want to leave Z till later, and this is exactly what this structure enabled us to do. So here on the left, I have an example of what a confocal image looks like for the vinculin rings. And you can see that they are quite clearly visible. They appear roughly round, and there's no real discernible structure apart from that. I'd just like to show you the results in fixed cells first. These were labeled with Alexa 488 and mounted in PBS with 100 millimolar of mercaptoethanol. You can, in fact, get much better blinking data than this, but we wanted to be able to show that the method could work in data where many fluorophores were overlapping in each frame. And it turns out that we were good enough to do a really bad experiment, which is exactly what was required. So what can we do with this information? Well, here's the wide field, and our reconstruction looks like this. And you can see it looks quite different to the confocal one. In particular, structures are apparent at corner points. In fact, corner points are apparent. The structures don't look round at all. They look roughly polygonal in most cases. And at the corner points, we see small strands of vinculin starting to nucleate, maybe hinting at nucleation of new podosomes. And I also wanted to show a second image. I'm not sure you can even see anything on the left. There's a very faint blob in the middle. And then on the right, we see a very small 240 nanometer podosome. Now, I thought it would be helpful to compare the sort of results you get with this technique to the results you can get with the other state-of-the-art super resolution instruments that are available. So we also imaged exactly the same structures using stimulated emission depletion. This is the Leica TCS SP5 system for anyone who's interested. You can see that, again, highly geometrical nature is evident using STED as well. And although it's slightly harder to see, at corner points you can also see thread-like structures again. And in fact, we also have structured illumination results, again on the same structure, vinculin, and on the left in two colors we have actin and vinculin. I'd like to thank Marie Welder and Brian Heinzman for this data. You can see, however, on the right, although the geometrical nature of these structures is evident, there are apparent slight artifacts occurring as lines across the image. And this proved to be quite important because we later went on to characterize the structure that we see in these images, looking at the binding angles that we see for the vinculin. So we reduced our images, we skeletonized them down to a series of lines, and then for each point where you have 3 or more lines meeting, we characterized the binding angles that we saw. Now, as you'll see, for both stead and storm, we saw a dominant binding angle. And the value that we saw overlapped well between the two methods. Stead, the expected resolution is 100 nanometers, and storm, the expected resolution for that data is around 30 nanometers. So you can see that, as expected, the storm results are somewhat more sharply peaked, but they do agree as to the value. But I'm sure you'll notice there's no sim results there. Now, the reason for that is when I processed the sim results, we did also get a very nice peak at a slightly different value. However, if you also processed a background area of the sim image, you got exactly the same peak. The artifacts that were introduced by projecting the grating and by the deconvolution process, in fact, produce a bias that's very strong if you're trying to detect any particular angles in those images. And I think it's an illustration of how with every super resolution technique, the interpretation requires caution. And of course, there are a number of other techniques, apart from our Bayesian one, which will also use standard type data in which fluorophores can overlap to boost the resolution. Maybe the best known one is the super resolution optical fluctuation imaging, or SOFI. And I carried out a comparison here. This is between the first version of the SOFI algorithm. There is a second version which is meant to achieve a slightly higher resolution. However, it's not freely available, and I had to write my own version, so I stopped after the first one. You can see the resolution does appear to be boosted from the wide field, but it is not boosted as high as 3B. However, of course, the calculation time is much lower, so which one you would choose would depend strongly on what you wanted to get out of the experiment. And secondly, Paul Selvin's work on shrimp. The resolution, I couldn't find any values in his paper, but I believe, looking at his images, it's around 100 nanometers. And it does require a very long image series. However, it only requires bleaching events. So if you only have bleaching, it can be a good approach. And finally, which I think is the most interesting result, really, we decided, before we submitted all this work, to just see what happens if we looked at live cells. Because, of course, standard fluorescent proteins also blink in live cells, and that information should be usable to reconstruct a localization image. So we used an M-cherry tailing construct at 37 degrees C, illuminated with a xenon arc lamp, and using a standard EMCCD on a completely standard wide-field microscope. And you can see the data. It doesn't look great, but if you stare at it obsessively enough for a really long time, you can see blinking events in there. And so we took this data, which was taken at 50 frames a second, and we split it into 200 frame chunks, and we performed reconstructions on those 200 frame chunks, and we got good enough data to be able to perform a reconstruction that achieved a resolution of 50 nanometers. And I think this was really fascinating. You can see it's maybe a bit hard to see. We have a whole cell there at the moment. There's an awful lot going on. But up in the top right for you, there's a podosome which here is starting out. We have 3 sides. It grows a 4th side, it grows a 5th side, and then it's taken back up into the cell. And let's look in a bit more detail at some of those formation and dissociation mechanisms. We identified that there were 2 distinct ways in which podosomes could dissociate, some of them unwound in a circle. They appear to also form struts across slightly across the circle to help with the protein trafficking. Or, as we see on the bottom, they can be drawn into the center. And I think even more excitingly, we also saw examples of podosome formation, and this was extremely different to what we thought it would be. We thought we would see winding out or going from the center. In fact, you appear to start from a strut across where the podosome will be. The podosome then blows up around the strut, and the strut is then, it disappears, it's taken apart. And finally, because I did think since I was in the neurobiology session, I should give you some neurons. I'd like to thank Tally Lambert at Washington State University, who has actually looked at neurons using this method. And he's looked at the dendritic spine in a fixed neuron and the microtubules in a fixed neuron, and he has seen a resolution improvement and images roughly what he expected. These were both done with bleaching only. So if you don't have blinking, it very much limits your resolution. So we think that this is really the lowest grade of resolution that we can get. But we thought it was quite promising because I didn't visit him or give him any indication about how to do the experiments. He just did the experiment and it worked first time. Though obviously now we think we can do much better, and we're very excited about where it's going. So I'd like to wind up by thinking a little bit about how super resolution is useful and the limitations, and how you choose what technique you use, because I think this is really the major limitation which is holding back use of it in research at the moment, this critical step of how do you choose the right technique to answer your problem. And so that each method has a completely different set of characteristics and is suitable for looking at quite different things generally. So with stimulated emission depletion, it can go quite fast. However, you do have to use higher intensity, so you risk phototoxicity in that case. It's not a very simple or cheap setup. You either need an expert to set it up for you, or you need to buy a very expensive microscope. You can achieve about 100 nanometers in resolution. You can use organic dyes generally. You can get away with standard fluorescent proteins. However, you do have to be a more expert user and have a different microscope to do so, and it will slow you down a lot. And you don't have to do any post-processing. Structured illumination, you get sub-second temporal resolution, but you're limited to 100 nanometers XY resolution. However, you do get a boost of a factor of 2 for Z. And you can use organic dyes or standard fluorescent proteins. You will be stuck with some post-processing. Palm or storm, you get hit on the speed with which you acquire data, down to minutes there. You can do it with a standard setup if you use organic dyes. And you can get excellent resolution and not much post-processing. So palm and storm are great as long as you don't want to look at anything in live cells. Sophie and shrimp are very nice compromise techniques. There are a few minutes to acquire the data, so similar to palm and storm, but the experiments are less challenging. They're less strongly constrained in terms of the data quality. You lose a tiny bit in the resolution, or a lot compared to palm and storm. You have quite a lot of flexibility on the dyes you use, and you're only looking at a few minutes. And then 3B, which is what we call the Bayesian localization of blinking and bleaching. You're down to seconds for the temporal acquisition. In fact, we're just now starting to push sub-second acquisition. It's very simple and cheap. You can do it on a standard wide-field microscope. Don't need anything special. It's 50 nanometers for your XY resolution. You can use organic dyes or fluorescent proteins, but you are going to be stuck with hours of post-processing, so there's always a cost. In conclusion, we think that we've invented a very exciting new technique of analyzing very dense data for localization results using a hidden Markov model. We're able to analyze data that has many overlapping fluorophores in it, which enables live cell imaging with a 4-second temporal resolution and 50 nanometer spatial resolution using non-photoswitchable fluorophores and a completely standard wide field microscope. We published it in Nature Methods. We're currently working up to the release of an ImageJ plugin. It's finishing its beta testing at the moment. And if you have any questions, do feel free to email me at the 3B email address. And finally, I'd like to thank Ava Roston, who I co-developed the algorithm with, who was absolutely critical in its development. He was of Cambridge University. Unfortunately, he decided to leave academia and is now setting up his own company. I'd also like to thank Rainer Heinzmann for help with the experiments, Gareth and James for access to Podosomes, Tijana Dillon and Jennifer Lippincott-Schwartz, with whom I collaborated on correlative work, and Rafal James and Anatoly, who I did some work on yeast with, and of course, finally, the Royal Society for funding me. And you all for your attention. Thank you very much.
In the live cell imaging, how are you differentiating between bleaching and blinking versus is there
At the moment, we don't. Because of the time scale of the podosome dissociation and formation, we don't think that a lot of movement occurs over the few seconds in which we take the image. Movement would also present to our algorithm as equivalent to switching of bleaching of that fluorophore and switching on of one next door. So the algorithm could cope with it. However, if we built movement into the model, it could then make it much faster in terms of the time scale at which we could resolve events. And so that's a current area of research for us.
So you have to make some assumptions about maximum movement speed and something in the next one.
Absolutely, and that is quite complex because obviously some structures move quite fast and some don't. So I think that, however, with the Bayesian, the hidden Markov model method we're using, because each variable only has to be weakly constrained, you only have to have a very rough guess at what's happening for each of them. We should be able to get some improvement by putting in some degree of modeling for the movement, even if it's not as accurate as we would like.
How close are the controls to optimal with respect to blinking or physical properties?
There is a lot of work in probe development. However, it's a bit of a black art. The process of blinking itself is not very well understood. For a long time, it was thought to be a transition to the triplet state. Now it's thought to be more likely to be an ionization process, or for the fluorescent proteins, a structural isomer. So because we don't understand the mechanism for blinking very well, it's quite difficult to optimize the blinking. For fixed cells and organic dyes, there has been a lot of work done on tweaking the reducing environment to optimize the blinking, and for that it's quite easy to control. However, in live cells, because you cannot tweak the environment too much before you damage the cell, I think it really will have to go with probe development, and that's really just, it's only just starting out, trying to find probes that will blink well in live cells.
To try to guide that development curve, do you have some sense of what stochastics you're looking for in order to facilitate the fastest possible acquisition?
I guess the problem is it's linked with what you want to look at. So it would be a different probe for each time scale. Basically, you want the time scale of your blinking to be roughly at the same time scale as you're acquiring a single frame. So the faster the process you want to look at, the shorter the frame time is going to be, and the shorter you want your blinking process to be. I guess the main thing that would generally help is getting probes with longer off times, because in general, all the probes that are viable in live cells, either you end up with a lot of overlapping fluorophores, or you need a very high intensity to drive most of the fluorophores into the off state. So finding fluorophores with longer off times at lower intensities would be an absolutely critical thing to trying to improve the data we get and the results.
So if I understand SOPHY, the SOPHY method, that's autocorrelation of single pixel data over a time series, and it's all local pixel operations. I don't have a good idea on how your algorithm, is it like a deconvolution? Do you have a point spread function in the calculation, or is it purely expectation maximization kind of?
It's really more like an expectation maximization, but we do build in information about the point spread function size and so on, but it's not really like a deconvolution. We're not, we are trying to ask, we're not saying, for this size of point spread function, tell me what you think the image looks like at high resolution. We're saying there are a range of possible point spread functions. Where do you think the fluorophores are? So it's asking a slightly different question to SOPHY.
How does the algorithm decide on the total number of fluorophores that it's going to locate?
So you have to start by putting in a guess as to the number of fluorophores. You can use the intensity in your image, and then you can calibrate using single fluorophores and get a pretty good guess as to how many you think there are. I mean, it can auto do that for you. It then, under the optimization process, will add or remove fluorophores. And one thing we recommend people do when they run the algorithm is to check that the number of fluorophores has stabilized, because if for some reason you get the fluorophore intensity very wrong, it can end up just increasing and increasing, and at that point you should stop and restart with a better calibration for the number.
Okay, thank you. Let's all go to lunch.